interesting talk just a while ago. Ms. Gandhita Kundaikar is an assistant professor in zoology discipline, School of Biological Sciences and Biotechnology, Goa University. She shall be the rapporteur for this session. I call upon our first speaker, Dr. Madhav Bilave, to present his work.
Good evening, everybody. Respected Chairperson, Co-Chairperson, Rapporteur, dignitaries of the dais, my fellow participants, and my dear friends. At the outset, I would like to acknowledge the organizers of the National Conference on the Animal Sciences, Integrated Solutions for Sustainability, for giving me an opportunity to come over here and present my work. The theme selected for the presentation is from the fish biology and the aquaculture, and the title for my work is Alterations in the Hematological Parameters of Freshwater Fish Sirinus mrigala after acute exposure to methanol. Yesterday, Dr. Sharma sir has very nicely given the deliberations of the genotoxicity, in which he has very nicely explained what toxicology is. If you look at the first few lines, you won't understand anything, which translates, okay, it is often condensed to the dose makes the poison. He has very nicely said yesterday that if you take the sugar, it can act as a poison. But if you take the venom of the snake in the small quantity, it acts as a medicine. So what it matters is the dose which makes the poison is very nicely said. Coming to the introduction of my work, toxicology is a discipline overlapping with the biology, chemistry, pharmacology, medicine that involves the study of the adverse effect of the chemical substances on living organisms and the practice of diagnosing and treating exposures to the toxins and the toxicants. The relationship between the dose and its effect on the exposed organism is of high significance in the toxicology. Factors that influence toxicology includes dosage, route of exposure, species, age, sex, and most important thing is the environment in which the toxicity is present. Aquatic pollution produced by the large amount of the waste material modifies the water in a negative manner. For the present study, we have selected the methanol. Methanol is an industrial chemical used since the 18th century. It is a toxic. Methanol is used to produce the biodiesel and also used in the ethanol manufacturing industries as the denaturative additives. With increasing production, transportation, storage, the possibilities of accidental release to the environment has increased. So for the present study, an attempt has been made to study the effect of methanol on the experimental fish Sirinus mrigala. Sirinus mrigala is one of the Indian major carps, which is a bottom feeder. And most of the toxicants, when they are introduced to the aquatic medium, they settle at the bottom. So the concept of selecting the Sirinus mrigala was there. The bottom feeder fishing, which will be exposed more to the toxicants, has been taken up at the experimental fish for the present work. The acute toxicity test for the 96 hours was carried out to assess the effect of the methanol on the selected fish species. We all know whenever the acute toxicity concept comes, it is of the 96 hours, and when we go for the chronic, it comes for the 30 days as far as the aquatic animals, especially the fishes, are concerned. The parameters selected for the toxicity, the hematological parameters, are utilized as the symptomatic apparatus for observing the poisonous reactions and the physiological status of the fish. The hematological indices included for the present study are the total erythrocyte count, total leukocyte count, hemoglobin, packed cell volume, mean corpuscle hemoglobin, mean corpuscle hemoglobin concentrations, and the last one is the mean corpuscle volume were taken into the account for the present study. Test chemical, as I said, is the methanol. Specifications as stated in front of you. I will not go into the details. Materials and methods. The total erythrocyte count by the Neubert's hemocyte. All these methods are for the 7 different parameters selected in the hematological parameter are the standard protocol of the experimentation for the aquatic toxicity. Coming to the results of our work. Table number 1, alterations in the hematological parameters exposed to the Sirinus mrigala after the acute exposure to the methanol. These are the observations. This is the graphical presentation of the same. I will not go into detail. When I will come to the discussion part of it, I will explain it in the very graceful manner. Coming to the alterations in the hematological parameters of MCV, MCHC, MCH, and TLC of the Sirinus mrigala after exposure to the methanol. And this is the graphical presentation of the same. This is a fish. Test animal was the freshwater fish Sirinus mrigala. And the test chemicals which we used in our laboratory was the methanol. And yes, we had the syringe. It was 1 ml sterilized syringe used for the blood sampling and the leukogram for the freshwater fish Sirinus mrigala to study the hematological parameters. Coming to the discussion part of it. In the present study, the first part is the total erythrocyte count. In the present study, the total erythrocyte count have decreased as a lethal concentration of the methanol in the Syrinus mrigala, followed by the various authors which have worked on the same parameters, taking the hematology as the concept, but with the different chemicals and with the different fish. Second part, total leukocyte count. In the present study, the total leukocyte count have increased as the lethal concentration of the methanol in the Syrinus mrigala, following other authors which have done on the similar work, taking into account the hematological parameters. Number 3, hemoglobin. In the present investigation, dose-dependent reduction in hemoglobin level was observed in the Syrinus mrigala treated with the methanol. Number 4, packed cell volume. The present study reveals the hematological profile, such as the packed cell volume decreased significantly in all the exposed fishes after exposure to methanol. Number 5, in the present study, the mean corpuscle hemoglobin value increased significantly on all the exposed fishes after acute exposure to the methanol. Number 6, mean corpuscle hemoglobin concentration. In the present study, mean corpuscle hemoglobin concentration increased significantly in all exposed fishes after acute exposure to the methanol. And the last parameter was the mean corpuscle volume. In the present study, mean corpuscle value increased significantly on all the exposed fishes after acute exposure to the methanol. And following are the workers which put in the chronological order. They have worked on the hematological parameters of different fishes. Now coming to the summary and the conclusion. In the present study, deals with the hematological impacts of the different lethal concentrations of methanol on the freshwater fish Syrinus mrigala. The work can be summarized as, in the present study, fishes exposed to acute concentration of methanol showed various changes in the hematological parameters of the freshwater fish Syrinus mrigala. Hematological profile, such as the packed cell volume, total erythrocyte count, and the hemoglobin level decreased significantly at acute exposure to methanol. The decrease in total erythrocyte count can be attributed to the inhibition of the erythropoietin. The decreased hemoglobin level caused reduction in the oxygen carrying capacity. Coming to the conclusion, the present study reveals that the methanol at various concentrations shows different mortality rate in the fishes when exposed. The alterations in hematological parameters in the freshwater fish Syrinus mrigala were due to the physiological responses of fish to the stress of the methanol. The study indicates that Syrinus mrigala, when exposed to the acute duration of methanol, was under tremendous stress, and parameters employed in this study can be adapted for further investigations as biomarkers of the damage caused to the aquatic organisms. Alterations in hematological profile confirm the sensitivity and severity of damage caused to the various tissues of the experimental fish, Syrinus mrigala, due to the toxicant methanol in the present study. These are some of the references which have been cited while preparing the presentation. And yes, a message, carry home, take home message. If you don't protect our earth, this is our future. And hope so, this is not comes in the reality. Though it's a virtual picture, hope in the coming times, this will not be the reality of our life in coming time. Thank you. Thank you. Yes, sir.
So you have used the methanol.
Yes, sir.
So how you just simply add the methanol?
No, sir. We have determined the LC0, LC50 values.
Okay. LC50, LC0 with the dose. I'm not saying the dose. How you add the methanol? Simply add the methanol.
Yes, sir. Methanol is miscible in the water, sir.
That is okay. My question is that if it is miscible in the water, so why have I taken the heat? Because it's going to affect much more in the heat other than the lab. Number one. Number two is that when you do the work, this particular hemoglobin and RBC count it varies.
Yes, sir.
Depending on the spawning.
Yes, sir.
Reproductive cycle.
Yes, sir.
So you have to conduct the same study with the different reproductive cycle period. Then you can also say how much methanol, which period, how exactly how methanol will create a shape for the sinus.
Yes, sir.
That's right.
With respect to you, sir, I would like to say that you have taken various parameters like the biochemical changes, enzymatic changes, and the hematological parameters. But for the presentation over here, I just stated the work which is based on the hematological parameters. We do have the enzymatic and acid phosphatase, alkaline phosphatase, and the various tissues like the gill, liver, brain, muscles, everything is there. Yeah, sir, it is a part of my project, sir. Yeah, it is based on the project, sir, sanctioned by the UGC. Thank you, one and all.
Any other questions, please?
Sir,
Yes,
thank you
please.
for the talk. Your last slide on summary and conclusion said you used the lethal doses to check the hematological parameters. Why not sublethal doses?
Madam, when we go for the acute exposure, we go for LC0 and LC15. When we go for chronic, we go for the 1/10 and 1/20th of the LC concentration. That is the sublethal concentration. If you want to expose the fishes for the longer duration, that is for the chronic, then we can go for the sublethal concentration. It is only for the 96 hours. There are a number of parameters, rather toxicants present. Methanol is one which we have selected because it was for my project. So the same thing I have taken up here.
Thank you.
Thank you very much, madam.
Any one quick question? From anybody from the audience? Yeah, I have a question. You
Yes,
have
sir.
not mentioned the sample size.
It's a fingerling size, sir.
Number.
So generally, when we go for the LC0, LC50, we take 10 liters of water and 10 number of fishes.
How many fish? The parameter hemoglobin, how many fish?
5 fishes were sacrificed and then average was taken, sir. Huh.
So you need to measure the size, everything, statistical parameters, to really understand this.
Okay, sir.
Now, methanol, when you add to water, isn't methanol volatile?
No, sir, it's a miscible component, so that's the reason we have taken up methanol,
It
sir.
is miscible, I know that.
Mm.
It is the most highly soluble ethanol or alcohol, but at the same time, it is volatile.
Yeah. I understand.
So whatever, that's why I asked how you are given the methanol.
Because over a period of time, your methanol is going to change, and you are also doing aeration.
No, sir.
You're not doing
No.
aeration.
In the experimental fishes, we don't aerate the fish. We at least
So
don't feed
wouldn't
the
that
fishes.
wouldn't that cause a stress on the fish?
No, sir. It is only for the 96 hours. We don't put the aeration. Before
There is a
acclimatization.
control? But I didn't see the bar charts there. Standard deviations you have done?
Yes, sir.
Because hematological parameters, they will vary, there'll be a
Definitely it
lot
will
of
vary, sir.
variation.
Yeah.
I don't think a conclusion can have
Vary, sir.
variation.
Yeah.
I don't think a conclusion can be drawn just on the basis of how much is the variation basically between control and your, methanol. I think it will have been better if you had shown enzymatic. That will have been more reliable.
That is also one more parameter which you
Yeah.
have taken, but not for the present this
Okay,
biochemical
fine.
changes, hematological parameter they have taken up, sir.
Fine. Yeah, thank you.
Thank
But
you very much.
methanol
Thanks, madam.
is volatile. You should have taken because over the time, methanol is
Yes.
evaporates.
Yes, sir.
Even if you don't aeration will generally evaporate faster.
Yeah.
But even without evaporation, without aeration, methanol will, I don't know, generally after 48 hours, the amount
Yep.
of methanol in the water might be very less.
Yeah.
What is the LC50 dose? Dose of an LC50.
Yeah, sir.
In how much water? In how much water?
Yeah, LC0 and LC50 values. Right now, I don't have here, sir. But we do have, sir, values, sir. You have determined the LC0 value, LC50 value. Yeah, and then these are called as the predetermined. Yeah, sir.
Look at the hemoglobin, look at the TC. Thank you.
Thank you very much.
Thank you, Dr. Bilavi. I now call upon our next speaker, Caroline Kanimoji.
So a very pleasant evening to one and all gathered here. So my name is Carolyn Kanimuri and I'm from Christ Deemed to be University and I'm pursuing my master's in forensic science. So today I'm here to present my paper on the topic rapid identification of species and determination of gender from tissue samples using real-time PCR. So moving on with a short introduction, as we all know, meat is a basic food product or an item that is consumed by each and every individual, let it be in India or worldwide, and it is mainly consumed only for their nutritional value, especially for dietary purposes. So these meat consists of chemical properties such as essential amino acids, proteins, fats, and micronutrients such as iron, zinc, selenium, vitamin B6, B12, choline, niacin, and riboflavin. So these meat products gains the color like red or whatever color like pale red, pale pink. So these are gained by the pigment named myo. Meat fraud as well as animal smuggling or trafficking is considered to be a detrimental crime that may lead us to any religious or ethical conflicts as well as in sometimes to legal concerns. So these meat fraud. So going on with the topic meat fraud, it is nothing but the meat is either adulterated or substituted with any lower quality of meat, and they when they are consumed by the people, let it be worldwide or anyone. So we also know there are peoples who consume a particular type of meat and not any all the types. So some may prefer only chicken meat, whereas some may prefer only beef. So there are specifications and also there are some religious beliefs during their consumption. So taking under all these things, so degradation in the authenticity of the meat is a main factorial or reason that may cause conflicts in the world. So as well as known in forensic aspects. Moving on with the forensic aspects, so these meat fraud is not only about adulteration, so there is also this cannibalism, which is known as consumption of human flesh. So in some parts of the country, people also consume human flesh as a meat product, and which are also banned by the countries, and many actions are also taken. So yeah, moving on with the PCR. So PCR is one of the DNA-based techniques. Even though various traditional techniques were discovered, it was known as non-reliable, and the results were tend to be inaccurate. So PCR was also known to be one of the DNA-based techniques, which is considered as a more efficient technique in species determination, and there are more advancements techniques are being discovered. So these identification of species from the meat and meat products is largely focused based on mitochondrial and nuclear genes. Yeah. Since mitochondria, as we all know, mitochondrial DNA is maternally inherited, and it is also established based on their activity that mitochondrial markers are very much useful in the species determination as well as the authenticity of species, the meat samples, rather than nuclear markers. So these mitochondrial genes have variable regions that are found in thousands per cell. So, for example, these meats can be consumed in various forms, such as processed meat or raw meat, or in sometimes they can also be consumed in a powdered form. So during these processed meat and minced meat, the meat is undergoing many kinds of chemical techniques. So where the quality or the authenticity of the meat gets degraded. So in such senses, it is people do think that it is unable to determine the species as there might be any kind of lack of DNA samples. So through PCR, it has been determined that mitochondrial DNA, even in lesser quantities, can also be determined. So it also enables the positive attainment of results where, as mentioned, the DNA, even when it is severely fragmented or it is damaged during the rigorous process of processed meat. So this study mainly defines about the species and its gender from tissue samples using real-time PCR and to prove its efficiency in species determination. So moving on with the methodology. So we have collected a total of 15 samples and it has been used in this study. So DNA extraction. The DNA extraction was mainly performed using Maverick nucleic acid extraction kit, where a 25 gram of each meat samples were taken, weighed, minced, and then was washed with 160 microliter, 100 microliter, and 20 microliter of lysis enhancer, and it was incubated at 56 degrees Celsius for 45 minutes. As after the incubation, a 20 microliter of RNA solution was provided in the kit itself, which was then added to each sample, vortexed once again, and incubated at 56 degrees Celsius for 10 minutes. After all these vortexing procedures, a 25 microliter of binding buffer was added and centrifuged for 1 minute at 4000 G which was then followed by transfer of supernatant 100 microliter of elution buffer and stored at 4 degrees Celsius under the downstill process. So after the DNA extraction, it was followed by the real-time PCR. So this real-time PCR for the detection of species, we have used two kits named as cow species identification kit and buffalo species identification kit, whereas the samples were especially collected from the cow and buffalo species. So the reaction was then carried out using CFX 96 real-time PCR system. So the procedure is nothing but a 20 microliter of the reaction, which also consists of 10 microliter of PCR enzyme mix, 1 microliter of primer probe mix. So here we used to use two primer probe mix where both are of Tachman probes, but they are of different things. So first one is in 1 microliter of primer probe mix of Tachman probe, it is specific for the mitochondrial DNA and XY chromosomal DNA. Whereas the second one of the 1 microliter of primer probe mix, the same Tachman probe is specifically used for the endogenous and control target DNA. And a 5 microliter of extracted nucleic acid and 3 microliter of nucleus free water was used. We also used a positive template control and a no template control for the validation of each assays. And followed by, as mentioned in the title, we also mentioned the gender determination. For the gender determination, the same 15 samples were taken and was determined using the kit called cattle sex determination kit. So here we have two probes, which is the VIC labeled probe and FAM labeled probe that targets the X chromosomal specific regions. So when the meat samples are identified for the female origin, the amplification to take place only at the VIC label probe, whereas for the male origin, the amplification should take place at both the probes. So here with a short discussion. So as we all know, there have been several issues regarding the meat fraud, and even in recent days, there have been misplacement or mixed of meat frauds. It can be not only through the adulteration or substitution, but they adulterate the meat, and the person who is consuming the meat is either affected by heavy medical conditions. Like these meat can be also adulterated using a powdered form of the meat. So there are various traditional methods also, like DNA based techniques as well as protein based techniques. So the primary objective of this study was to determine the species and the gender. So these are the results. Out of the 15 samples, it has been determined that 2 were of buffalo species and 11 were of cow species, whereas 1 were of mixed species of both cow and buffalo, and 1 was based on the gender determination. The 12 species were determined to be of female origin, and the 2 species were determined to be inconclusive, and 1 were determined to be male species. So as you can see the spectrogram, the first image depicts the amplification, which determines the specification of cow mitochondrial DNA, whereas the second image determines the amplification of both VIC and FAM labeled probe. So these were the number of samples and the data. Yeah. The top image, it determines the presence of mixed sample where you can see both the cow and the buffalo species. And the left image, which is seen here, it depicts about the X chromosome specific VIC labeled probe where you can see the presence of X chromosome DNA only, and you can just identify the green line present in the image, which depicts the presence of female cattle origin, whereas in the last image you can see the presence of VIC as well as FAM probes, which determines the male cattle origin. Study, we conclude that the origin and the species can be determined. It is also concluded that real-time PCR is an efficient technique, which is also fast, reliable, and accurate, and is also a technique for the species determination. Thank you.
Yeah, quick questions.
Yes, sir. Yeah. Yeah.
Meat fraud can be used for or using the certain chemical formalin or this
Yes, sir.
adulterations. So only this PCR will not able. How will detect the PCR techniques to such chemical adulterations are there on the meat or not? Mixing meat is a different story. You can able to get RT-PCR is a DNA marker. You can able to do it, but other way how you can do the meat fraud?
Other than
PCR will not going to help, na?
yes, sir.
Then what is it?
So, sir, to say like there are different kind of techniques is also going.
Fraud generally, what is the market meat fraud? Trading meat fraud is not only the mixing.
Yes, sir.
Mixing is not that way. Mainly, it is been by adding some chemical or something.
Yes, sir.
So the forensic this thing has to be take care of the what type of mixing is there.
Yes, sir. Based on the chemicals or any kind of foreign particles present in it.
Yeah, any other question, please, from the audience? Yeah, okay, thank you.
Thank you.
Thank you, Ms. Caroline. Our next speaker is Dr. Aditi Naik.
A very good evening to all. Today I'll be speaking on the research work which was conducted as part of my PhD program entitled Anti-Tumor Efficacy of Annona muricata Extracts Against Ultrajacitus Carcinoma and Dalton's Lymphoma Ascites, Facilitated Tumors in Swiss Albino Mice. Enriched in nature is all that is used for well-being of humankind, especially to fight against various diseases and disorders. And amongst the various plants that have been used for the purpose of medicine, Annona muricata is one such plant which is known for its therapeutic and beneficial attributes for the various organs that it possesses, namely sorry. So this plant is been known for its curative and beneficial attributes for the various organs that it has, the rind, pulp, seed, leaf, bark, and root, which was studied for my PhD program. Now, this particular plant is known for the phytochemicals which are present in this particular plant group, namely the alkaloids, the phenolic contents, and most importantly, a unique class of phytochemicals that is acetogenins, which is attributed for its anti-cancer curative benefits. Extensive research has been carried out on Annona muricata with respect to the phytochemicals present in various organs of the plant, the pharmacological activities, the cytotoxic activity with regards to various solvents that were used for extraction of these phytoconstituents, the toxicity studies. But however, we found that there were certain research gaps, and that was the comparative phytochemicals which was not being profiled in different organs of Annona muricata using various spectroscopic techniques. The antioxidant and the cytotoxic activity of the various organs were also not evaluated, and thus we focused on evaluating the various organs of Annona muricata, particularly from Goa region. And from the various solvents that we tested, we found that the methanol extract was showing a plethora of phytochemicals which were extracted from different organs of Annona muricata. Hence, as a preliminary study, we screened the various cell lines, namely MCF7, HeLa, A549, and oral cancer cell lines for the anti-cancer activity. So also we studied these cell lines using SRBSA and MTTSA in order to understand their cytotoxic activity. And amongst the various organs tested, we found that the leaf extract was showing better efficacy towards cancer. Further, we also evaluated the genotoxic potential of the various plant organs, more particularly the pulp and the leaf extract, and in comparison with annonacin, which is a standard acetogenin which was used in my study. Having said that, today's talk will be focused more on the anti-tumor activity of Annona muricata, and this is a representation of the work that has been summarized with regards to the in vivo study that was carried out using the EAC and the DLA cell lines. in the mice. Here we studied the acute toxicity study of this leaf extract using hematological, biochemical, and histopathological studies. We also did in vitro cytotoxicity studies, and further on, we did the in vivo anti-tumor activity using ascites tumor and solid tumor ascites carcinoma in comparison with the standard drug cisplatin. Here we grouped the mice in 5 different categories, where 6 mice were studied in each of the categories, where the group 1 mice were administered with was a negative control in our study. Then the group 2 was the mice which was bearing either of these two cell lines along with a dose of 3.5 cisplatin. Then we used 100 mg per kg of limb extract, that is the leaf extract of Annona muricata, followed by 200 and 500 in an increasing dose. So these are the results of the in vivo anti-tumor activity of Annona muricata leaf extracts. And in this particular study, we found that when compared to the normal the mice, we saw that there were not many much hematological differences that were observed in the various parameters that were evaluated, except that the WBC count was increasing. The hemoglobin count was found to be decreased, but that was not a comparatively larger difference that we could notice with respect to the control group. Further on, we evaluated the biochemical parameters in the control mice, and the mice which were treated with this particular leaf extract at 500 mg per kg body weight of the mice. And again, we saw that there were not much differences that were observed, drastic differences that we could observe, but yes, there were minor significant differences that could be observed when administered with the leaf extract of Annona muricata. These are the histological sections of the kidney, liver, spleen, and brain of mice in the acute toxicity study that we evaluated. And here again, we saw that there were no histopathologies evaluated or noticed in this control group, as well as the mice which were administered with this particular extract of Annona muricata. Further on, the in vitro cytotoxicity was also calculated. And here we saw that based on the IC50 value, the extract was showing more cytotoxic activity towards the Eldridge ascites carcinoma compared to Dalton's lymphoma ascites. These are the images of the mice. The A represents a mice which is a control mice which has not been administered with any drug. B is a mice which was not given any, which is administered with the cell lines, that is the ASE and DLE cell lines, which shows signs of tumor. CDE represents mice which were administered with 100, 200, and 500 mg per kg of the Annona muricata extract. And F is the mice which was given the cisplatin, which was used as a standard drug for comparison's sake. So based upon these studies, we found that the survival time of Eldridge ascites carcinoma bearing mice, we saw that with increasing concentration of leaf extract, we saw that the survival was increasing. And we saw that in comparison to the control, when the mice were administered with 500 kg of this 500 mg per kg of the leaf extract, we saw that there was 50%, 51% increase with respect to the life span. Further on, when the cisplatin was administered, we found that there was 56% increase in the lifespan, which again shows that the extract that we administered is showing some beneficial attributes to this mice which were harboring these tumors. So also we studied the solid tumor models in the mice, and again we saw that with increasing concentration of the leaf extract, the tumor was also declining. And we can see that with respect to the control, when the 500 mg per kg of the extract was used, we could see that there was almost 65% decrease in the tumor volume. And we could see that from the control, from the zero state, we could see that there was almost 6% increase in the tumor volume compared to that of the control, where there was almost 15% increase when the mice were when the tumor was calculated at around 42 days of tumor of the mice which were administered with these tumors. Additionally, we have also studied the MCF7 cells and the cytotoxicity study, which is in support to the previous study, wherein we saw that the proliferative activity or the antitoxic activity which was exhibited by the leaf extract of Annona muricata with respect to MCF7 cells, that is the breast cancer cell lines, is leading to cell cycle arrest and programmed cell death through the involvement of ROS and upregulation of caspase-3 signaling pathway. To conclude, our study demonstrates that the potency of Annona muricata leaf extracts, which shows in vitro and in vivo anti-tumor activity. The toxicity parameters evaluated did not show any significant variations, which suggests that there was no alteration in the general state of the mice after administering with the Annona muricata extract. Further, this extract exerted dramatic decrease in the cell viability and increased protection against the experimental animals. And in comparison to the different concentrations that we tested, 500 mg/kg of body weight of this particular dose regressed tumor growth, viable tumor cell count, increase the overall survivorship and life expectancy in tumor harboring mice. The findings thus imply that this extract may potentially contain better bioactive metabolites with significant anti-proliferative capacities, making this extract a better dietary drug for prevention and metastasis of cancer. To support the above, further investigations are also being carried out to elucidate the exact mechanism of action and its metabolism studies. Thank you so much.
Thank you, Dr. Naik. Just any one question from the audience?
So the bioactive compound, we took the crude extract, sir. Bioactive compound, so we did the genotoxic study only of annonacin, which is a bioactive compound, which is copiously found in the leaves of Annona muricata.
Okay, thank you, Dr. Anita. Next person, please.
The next speaker is Sarita Rebello. Good evening, everyone. I'm Miss Sarita Rebello from Zoology Program, School of Biological Sciences and Biotechnology, and I'm here before you to present on the topic carbon black nanoparticles provoking turmoil in mus musculus. Nanoparticles, they exist naturally as well as a consequence of human activities. And specifically, carbon black nanoparticles, its exposure is increased to the humans as well as the environment due to the incomplete combustion process. And these particular nanoparticles in itself, they are not toxic, but due to its ability to mingle with the cellular level, its agglomeration property and low clearance rate, they can be toxic to the human. Now, these particular nanoparticles, though they are very small in size, they pack a big punch because of their applicative domain, because they are used in different fields like manufacturing of the tires, plastic pipes, plastic wraps, then majorly in ink production, batteries, and photographic films. Now, a multitude of research studies had been done wherein these particular nanoparticles have shown lung toxicity, pulmonary toxicity via intraperitoneal, intratracheal, and inhalation processes. But there was a gap in which there were no majorly chronic studies done wherein oral route exposure of these nanoparticles was used. Hence, this particular study revolves around four major objectives wherein we are going to understand the toxicity of these carbon black nanoparticles on the behavior, anatomy, morphology, renal and liver functioning of the mice. Going on to the methodology, A total of 24 mice were divided into 4 groups, 1 control and 3 experimental, of which the experimental group mice were dosed orally or, with 3 different doses, that is the 5, 10 and 20 mg of carbon black nanoparticles per kg of their body weight for a continuous period of 30 days. And after this particular 30 days exposure, the mice were sacrificed via cervical dislocation. The kidney, liver was isolated along with collection of blood and urine. And all these tissues were then further processed for histopathology as well as biochemical estimation. Coming on to the results, in behavior, an increased rate of rolling of tail as well as hyperactivity was seen in the mice, which might be due to the effect of these carbon black nanoparticles to the central nervous system, changing the hormonal release or the release of the neuropeptides. And in morphology, distinct hair color change was seen, eye color change was seen, as well as hair loss was also noticed. Now, this could be due to the carbon black nanoparticles draining via the circulation either into the epidermis or affecting the lens, retina, or even the optic nerves. Apart from that, a significant decrease in the body weight of the mice was also noticed. Coming on to the anatomy, in the histopathology of the liver, there was distortion of the hepatocytes along with distortion of the sinusoids, which was observed. And in both liver and kidney, there was clustering of the inflammatory cells, which was also noticed. Specifically in the kidneys, distortion of the glomerulus and the tubular epithelium was more evident. Different types of metabolite were estimated to conclude with the liver physiology. For example, carbohydrates, free sugars, total protein, albumin, total cholesterol, triglyceride, reduced glutathione. These were all assessed and which showed significant increase in their concentration, along with bilirubin, which also showed significant increase in its concentration, which could be due to disruption of the sinusoid due to the carbon black nanoparticles. And the two major liver damage marker enzymes, that is the ALT and AST, were also assessed, which showed a significant increase in their enzyme activities. Whereas the catalase enzyme, which was there, that shows a significant decrease in its concentration, be due to overproduction of the ROS, which in turn had an inhibitory effect on the catalase enzyme. Now, similar results were also observed in the kidney physiology test, and specifically urea and creatine, both in the blood as well as in the urine, was seen to show significant increase. Particularly was or can be due to the glomerular destruction which was caused due to the effect of carbon black nanoparticles. Now, any study becomes significant only when it is beneficial, when it has an applicative domain, and when it has some future explorations. So coming to the applicatory domain, this particular study provides a repository of carbon black nanoparticles toxic effects, which will give us or a lot of industry peoples to come up with some preventive measures that will cut down the effect or the exposure amount to the common layman as well as the industrialist. And for us as customers or consumers, it will help us to be aware about the detrimental potential of these carbon black nanoparticles. And in future exploration, we can do in-depth study about the genotoxicity, neurotoxicity caused by this carbon black nanoparticles, as well as this carbon black nanoparticles can be engineered again to change the surface modification by adding different types of compounds which might decrease the toxicity of these nanoparticles, and hence further the engineered nanoparticles can be used in manufacturing of the different types of products. Now, in conclusion, how is this study useful for us as laymen? I will love to summarize this with few example. For example, the pen which you all are using, the ink which is there in that pen, it has carbon black nanoparticles, and a lot of time we tend to write on a hand with the pen just to remember some things, or by mistake also the ink from the pen comes to our hands, and that is how by mistake sometimes without washing the hands we tend to eat the food. So we tend to orally gavage or orally eat the ink which is there, which is containing the carbon black nanoparticles along with the food. Apart from that, newspaper are one of the most famous food packaging material. And these food packaging material are used locally in different stall to wrap samosas and different oil products, which from the newspapers, the ink will get absorbed to the food and that will be consumed by us, directly settle on the food. And that is how we consume these particles via our food. So if carbon black nanoparticles being so small in size can pack a big punch of toxicity due to their agglomerative properties, low clearance rate, then we as human beings having so many nanoscale entities can pack a much bigger punch to eradicate this toxicity by being a good consumer, being aware about the products we use, and changing our consumer habits. Thank you.
Thank you, Ms. Sarita. The conclusion that catalase decreases because of ROS is wrong. If ROS increases, SOD and catalase have to increase. It cannot decrease.
So, but the overproduction.
Overproduction. That's why basically SOD and catalase, they are supposed to scavenge the reactive oxygens, ROS. So they will not affect the SODs and catalase there. They are supposed to take care of ROS. So ROS increase is your conclusion that it is decreasing or inactivating. So that is not correct. Catalase and SODs are their job is that.
Okay.
Okay. And another thing is, everything is blue. You can't make out what is what. Your bar charts. You could because then it becomes slightly, because I was getting confused what is what. Anyway. Yeah. Okay, thank you.
Okay, sir.
One small query. Do you have any information about the tracking of the carbon nanoparticles in system biology?
In system biology.
System biology, tracking of the carbon nanoparticles in the system biology, because these are so small. What is the size of the nanoparticles you are using?
Less than 100 nanometers, sir.
So very small, they may enter in the track of the system biology. So is there any information that how they are taking or entering in the path? You have shown that in the glomerulus, it's there.
Yeah, sir.
They are going in the ultrafiltration. Are they coming the reabsorption track and other things? So any information about that?
Yeah, sir. There are some molecular studies done which says that this particular carbon black nanoparticles, they distort some cellular pathways, and that is how they enter different membranous structures. For example, they affect the MAPK pathway, that is the mitogen activated protein kinases, and through that they enter the different types of system. This is the.
Are they coming in the ultrafiltration or not? In the ureter?
Yes, sir.
They are coming.
Yes, sir.
Means they have been diagnosed in the sample of the urine once the carbon nanoparticles are given to the organism, and these are collected in the urine sample. Is it true?
So not in the urine sample, but in the histology, the accumulation is being noted down, sir. Yes. Sir? No, so this was my MSc dissertation work. Yes. Dr. Sharma.
Yeah. Okay, next presentation, please.
Calling up our next speaker, Dr. Avelino Da Costa.
Good evening, everyone. It's not my work, it's a work of my dissertation students I'll be presenting over here. Determination of microplastics in economically important organisms of Goa. And I'm not here to compete with any of the other participants over here, just to share some of my research work. Since I am a part of the organizing committee, I just thought it would be the right time to do so. So just to give an introduction, I mean, it's already spoken about by Dr. Pradeep and also by Dr. Shamila Montero about the coastline of Goa and how seafood is a very, very important resource for the people of Goa. And you can see, as evident by just looking at these pictures over here, I'm sure some of us must be salivating looking at these images, okay, because these are some of the dishes that we most frequently like to consume, you know, in the form of curries or in the form of rava fries or, you know, any type of preparation of these seafoods. Okay, so Goa does, produce. When I say produce, I mean we do a lot of catching of these fishes and these other organisms, okay, for consumption. And definitely it is obviously going to be the staple diet of the people which are living along the coastline. So coming to the pollution status of Goa, even though, like, you know, we are harvesting so much from the aquatic environment, the coast of Goa is also considerably polluted by a number of pollutants. So there are a number of reports which are available, such as in the presence of heavy metals which are there, the presence of petroleum hydrocarbons, pesticide residues, and also recently also has been seen that even plastic has been found in not plastic was always there in the waters. I mean, people are always dumping in the waters, but more specifically the microplastics or the nanoplastics which are being found, especially in the tissues of the organisms. So how these substances, how these substances enter into the waters is basically from the anthropogenic activities which are being carried out, okay, especially in case of mining activities, from, you know, the barges, from the upstream mining activities. Then tar balls, as you all must, in the second image over there, you can see the black deposition on the beaches. This is coming from the tar balls, basically, which are weathered crude petrol, okay, which has been in the water for a long time and become weathered in the sun and wave action and has gotten deposited on the beach. Many people think it's clay, but it's actually tar balls, okay, and when you smell it, you get a distinct petroleum smell to it. And then obviously plastic is there because you see a number of people still dumping garbage into water bodies, thinking that, you know, they are cleaning their houses, but dumping the littering the environment. So a number of these practices are still being seen. It's very sad to see that even some of the school children, I see that they are still littering on the roads, even though, you know, they are being informed about not to litter, keep your areas clean and everything of that sort. So what are microplastics? Microplastics are any type of plastics which are less than 5 millimeters in size, and they come in different shapes and sizes, they can come from various sources. Also, they can come from fishing gears, they can come from the sewage, drainage, textile, cosmetic industries, face masks, etc. And these microplastics are basically going to be formed from weathering processes. So larger size plastics can get broken down over a period of time due to UV rays, and they get broken down into these smaller size plastics called microplastics, and these can easily enter into the into the gastrointestinal tract, or, you know, can get lodged in various body parts of organisms. And different types of plastic types we can get, such as polyethylene terephthalate, polyvinyl chloride, polyethylene, and so on and so forth. So I'll not go through the review of literature. It's just that microplastics have been found in various organisms throughout the world, and a number of toxicity studies also have been carried out in them to try to find out what exactly is the toxicity of microplastics in them. And the significance is that in Goa, very few studies have been done with regard to microplastic, because we are a largely seafood consuming state, and we need to know what's the status of these microplastics in our Goa. Okay, so I've selected two organisms, which is Meretrix casta and Lithopenes vanumi. So Meretrix casta is this bivalve, which we call as Tisrio over here, and it is found in the intertidal zone, and it can easily be collected, and it is consumed by the local people. And the other organism is Lithopenes vanumi, which is referred to as a white-legged shrimp, which we call as sungta over here. And these also will be can be harvested, and they will be used for consumption. So the study site, I've just combined two of my dissertation students' work. So one student had collected the bivalves from Siridam beach, and the shrimps were collected from a sluice gate at Siolim. So these were the two sites from which the organisms were collected. And what we did was, from the bivalves, we took the whole soft tissue and we digested it. And in case of shrimp, we use the hepatopancreas and gastrointestinal tract, and we digested it with 10% KOH and 30% H2O2, which is the standard protocol that is used. I forgot to put the reference over here, but it's a standard protocol which is used for digesting the tissue to obtain the microplastics. There are other procedures also where they use acids, but acids will degrade the microplastics also, so you will not be able to, correctly analyze what type of plastics are there. So then we filter this solution through a 0.5 micron filter using a vacuum filtration, and then we dry this filter paper and we observe it under the stereo microscope. So these are some of the observations that we saw in case of bivalves and in case of shrimp. So we found a large number of fibers, especially in case of shrimp, and we found a lot of fragments in case of bivalves. Okay, and these fragments, so the difference between films and fragments, films are very thin membranous type structures, whereas fragments are a little harder structures. Okay, and you can get them in different colors also. There are different shapes which you can get of them. So we actually, the point of this is that we have to count the number of plastic particles which we find on the filter paper, and that will give us the value of how many microplastics were present in that particular organism. So we also went ahead in trying to determine what type of plastics they were using micro-Raman spectroscopy. And this is just one example where we found a polyethylene curve, where we saw one of the fragments was of polyethylene. And so we also found, I didn't put it up here, but nylon also we observed. Okay, nylon, even polystyrene, we got the spectrums of these. And these are the results. So you can see that in case of the bivalves, we found a large number of the fragments compared to the fibers, but in case of the shrimps, we found large number of fibers compared to the fragments. So why this must be the case is because mostly in that sluice gate area, they release the nets over there. So mostly the nets, when they get degraded, the fibers will be entering into the waters. And so these shrimps must be taking up these fibers to a larger extent, whereas the bivalves are more of the filter feeders, suspension feeders. So I'm just giving that conclusion, but it has been supported by other literature as well. So this is one logical conclusion which I could think of. And this is to say that microplastics are found in various other organisms also. And so what we have done also supports our study. And other reports have also said that, these microplastics are coming mostly from fishing sites, from fishing gears, I mean to say, from discharges, from garbage and all of these things. And so it is definitely, if this situation is there in Goa of pollution, it will definitely be there in our seafood as well. So our conclusion states that we found a lot of fibers and fragments and films in microplastics in their tissues. And this could be because of effluent discharge, extensive fishing activities, discarded nets and ropes, etc. And because of this, there will be some type of bioaccumulation and biomagnification taking place in the food chain, which could then be problematic to humans in the future. So these are my references, and I'll just end with this slide over here again, showing you some vegetables are there, but
Dr. Thank you, Dr. Yeah. Any?
Yes, sir.
Microfibers means how long do they stay in the environment? When these are degraded, then ultraviolet radiations may break them up also.
So they break down even further, sir. So they remain for a very long time. Their half-life is very, very long actually in the environment. So to actually get the correct age of that plastic would be very difficult because they just keep on breaking down. So even if it's a small particle, it doesn't just dissipate. It breaks down further into nanoplastic then.
Yeah.
So there's no end to it. Nanoplastics are even worse than microplastics.
And there is no way by which these can be eradicated by any bioremediation.
So there are, I think my colleagues from biotechnology and microbiology are working on some strategies where they are using bacteria to try to break down these microplastics.
Fungus or bacteria?
Yes, sir, yes, from the aquatic environment.
Sir, one small question.
Yes, sir.
Why Pacific white leg shrimp? Litopinus vanami has taken because it is not from origin. It is a cultured species. Yeah,
it is a cultured species, but it is a largely cultured species in Goa actually. So most of the shrimp farming areas, they culture Litopinus vanami because it is a very hardy species, very sturdy species actually, and can survive a lot of changes in salinity, pH, and all of these things. So it is a widely cultured species. That's why I selected it. I know it is not a native species, but it is being harvested to a large extent. So there will definitely be some anthropogenic effect even on these shrimps as well.
Yeah, since it is a culture species, now even in the pond, we use the polythene sheet.
Yeah,
inside the pond.
yeah,
So
yeah.
it is inside the plastic only. So
Exactly.
plastic getting into it is
Exactly.
common.
I've seen some where they are using these plastic sheets to even allow the shrimps or the fishes to lay their eggs on those plastic sheets over there in some fishery, in some, I don't know which farms, but they're using the plastic sheets also. So whether that is going to have an effect on, the fish that we get from there, I don't know.
Thank you, Doctor.
Thank you,
Next one,
sir.
please.
Our last speaker for the day is Prachi Talavnikar.
Good evening, all. So, I'll be talking on the topic eco-landscaping can be the best choice for biodiversity and beautification of Goa University campus. The work is being carried under Dr. Minal Desai, and a part of work is contributed by Aishwarya Nini. Landscaping is an important aspect of beautification of any property. Greening and landscaping of institutional campus also grants point for National Assessment and Accreditation Council. But landscaping leading to alteration of any habitat without undertaking serious impact assessment may be the cause of loss of exclusive flora and fauna. Since birds are good biological indicators, the study focuses on the bird diversity and its association with different habitats on Goa University campus. Goa University campus is located on the lateritic plateau with many institutional buildings and its unique biodiversity. So on Goa University campus, we selected different sites, 5 different sites with different habitat. So the first site was a forest fragment, which is a partial residential area with semi-forested area, and the vegetation principally included grasses, shrubs, and trees. The second site was rocky patches with shrubs, which is a area near the faculty quarters and HRDC building with an open lateritic rock patches, and the vegetation included grasses, thicklets of shrubs, and trees like Bombec ceiba. The third site is an open field with very less natural vegetation and which is under high constructional activity. Site D is a shrubby area near the library with large number of shrubs, grasses, and open rocky patches. And the last site is a fragmented patch with open lateritic rocks with grasses, shrubs, and trees on the periphery. So the study was conducted from September 2022 till February 2023 at 4 nightly intervals from 7:30 a.m. to 10:30 a.m. using the point count method. In the present study, 21 new bird species, including 8 migrants, were added to the checklist of the Goa University campus, thus making it with a total of 143 species. Among these, 15 bird species are found to be breeding on the campus. This is the list of the 21 new sightings of birds on the Goa University campus. So, these are some of the bird images I could capture on the campus, including the coppersmith barbet, the gray-breasted prinia, chestnut-tailed starling, Tickle's blue flycatcher, blue-faced mulcoha, it is a juvenile, Tickle's leaf wobbler, scaly-breasted munia, and thick-billed flowerpecker. So coming to the breeding of campus, a total 15 species of birds were found breeding on the campus, and the two raptor species were Brahmini kite and white-bellied seagull. Earlier studies showed total of 10 species that were breeding, and in the current study we found 5 more species that are added, including the scaly-breasted munia, barn swallow, and crimson-backed sunbird. A total of 51 nests were recorded on the campus, in which the crow nests were the maximum, with 32 number. This is a picture of a Brahmini kite nest and a crow nest. When comparing the bird species in the 5 different sites, the maximum were found on site B, which is a rocky patch area with shrubs, with a total of species. On site A and site E, a total equal number of species were recorded, with both of which were fragmented areas. And the list were recorded on site C with 23 different species. On site A, which was a forest fragment, 34 species were recorded, and the exclusive species here were the Indian grey hornbill and the Malabar grey hornbill. The highest population of recorded was of crows and jungle myna, in which 16 crow nests were recorded. On site B, which was a rocky patch area with shrubs, a total of 62 species were recorded, which was the maximum among the 5 sites. In this, the migratory species, the rosy starling, were the most dominant species, which were generally found on the red silk cotton tree. To conclude, Goa University is a Goa University has a unique habitat with 143 bird species and also a breeding site for habitat-specific birds. Sustainable solution is eco-landscaping, which draws on the principle that landscape ecology to create that evolve and sustain themselves over time with limited human intervention. In end, I would like to say that habitat-specific species of flora and fauna is equal to the natural beautification of campus, which means retaining the natural flora and fauna in its natural habitat will lead to the beautification of the campus. These are the references, and thank you. No, sir, I'm doing internship right now, and it's that. No, sir, it is a. I have joined for internship now, for last 6 months. Internship work, sir.
Look, first and foremost, when you do for field work,
Yes.
you cannot simply camera. Nobody will accept it. You have to use all the photographs with the GIS station, date and time.
Okay, sir.
Otherwise, I can show any photograph. I can Google. I can take so many photographs.
Yes, sir.
Okay.
Yes.
You should not do the mistake.
Okay. Yes.
Second thing I have just simply question is so that because you show a table the 10 years gap and the first, second and third gap. 10 years data.
Yes, sir.
So from this table you have so that this time you saw the 21 new added species.
No, sir. I think.
Yeah, only 21 some species is only added.
Yes, sir.
That means, but how many species is not there earlier reported? Some species disappear. Occurrence of some bird disappear, which were observed earlier 10 years back.
Yes, sir. The grey hornbill.
How many species are
2 species. Okay, sir.
You are highlighting a new species, that means new observations.
Yes.
In this area, but you have to say that what was observed earlier is not also come now.
Yes, sir.
Then second point, if you are doing, then you have to say why.
Okay.
Why these things are happening.
Okay.
Then you saw the different citation side.
Yes.
You saw some difference.
Yes.
Why the difference is there? That should be also taken into consideration.
Okay. Okay, sir. Yes.
Otherwise, it will be simply watching the bird.
Yes, sir.
I can also watch the
Understood.
bird, na?
Yes.
But I cannot be the only one.
Yes.
So these are the things that you take care. I think you can do the good.
Yes. Thank you. So I missed that point. So it's like for the reason of greening the campus, the plantation drives are carried out. But without a serious habitat assessment, the entire ecology can change. For example, the acacia plantation, they can change the entire ecology of that particular habitat. So before, doing any development or these plantation drives, the habitat should be accessed first.
than your mineral madam actually. That's why I was more curious to understand what is that no plantation drive. I think you have put that point very clearly.
Yes.
What do you mean by?
Yeah, the grassland should be retained as a grassland, not should be converted into a plantation or a forested thing. So,
Because now there is a lot of things happening.
Yes.
I think Mineral madam is aware about it, but I'm just pleased that I was not clear with no plantation drive. So whether we should do plantation or not to do plantation. That is the question.
Yes.
You are right. You are justifying the point.
sentence is not proper. The point bullet should be properly defined.
Okay.
This thing. You have taken 5 sites.
Yes.
So you have the entire area of the Goa University landscape.
Yeah.
So are you planning to this thing systematically analyze the entire landscape?
Yes,
And
sir.
which will be the no development zones if you are considering future
Yes, sir.
conservation?
Yeah.
So that this data will help you in analyzing which area can be set aside,
For conservation.
which should not be
Yes.
touched or should not be converted into plantation, as you
Yes.
are rightly saying. Okay,
Yes.
good, thanks.
I have got a few things to add. First thing is GPS location, what he has suggested, it should be there.
Yes.
Then you make GIS inference. You know GIS?
Yes.
Geographical Information System
Yeah.
for the prediction. And here this no plantation drive and other thing would come into consideration. Next is you should have IUCN status of these.
Okay.
Actually, what is the status of these birds? Maybe 1 or 2 species are really under the threats or in the endangered category or whatever.
Mm.
That can give a good value to the campus, that this is the campus that is maintaining these species which are under the threat or close to extinction or whatever status. So IUCN status
Yes.
at the campus.
Yes.
And the last, which was I was also discussing with your supervisor, Meenalji, that recording the videos, getting the audios, and preparing the spectrogram with reference to different behavioral patterns. So this will become a science, good science.
Yes.
It's a good laboratory. It's a natural laboratory where you
Yes.
can do all these things.
Yes.
Thank you.
Yes. Thank you.
So this was the last presentation. So we had a 6 presentation, starting with Mr. Madhav Bilave, who spoke on the alteration in hematological parameters of C. mrigalla after acute exposure to methanol. So this was followed by Caroline Kanimozi, who spoke on the identification of species and determination of gender from tissue sample using real-time PCR. And then we had Dr. Aditi Naik, who talked on anti-tumor efficacy of Annona muricata extracts against certain cell culture lines. And then we had Sarita Rebello, who spoke on carbon black nanoparticles and their effects in mus musculus. Then we had Dr. Evelina Di Costa, who spoke on microplastic contamination, basically in important organisms of Goa. And this was followed by Prachi Talavanekar, who spoke on eco landscaping in Goa University campus. So the presentations were good. So I think we'll be tabulating the marks and handing over to the organizer. Thank you very much. Thank you, the organizers and other members, for giving us the opportunity. Thank you.
Thank you, sir, for the concluding remarks of this session. I would like to request Dr. Meenal Desai-Shirodkar to present a token of appreciation to our chairperson, Professor Sanjeev Ghadi. Requesting ma'am to present a memento to our co-chairperson, Dr. Pradeep HD. Also requesting ma'am to present a memento to our rapporteur, Ms. Gandhita Kundaikar. Thank you, ma'am. We have amongst us Shri Ajay Saxena, retired Chief Conservator of Forest. We thank you, sir, for gracing this occasion with your presence. We've come to an end of this session. We shall now break for a quick tea in the cafeteria, following which a short sightseeing program has been arranged for everyone and for the delegates, and the bus is already outside the auditorium. Thank you, everyone.